Hi, so I'll be presenting on my project for my PhD on Facemap. And so I'll be presenting some of the background as to why we started working on Facemap and tell you a bit about the tool and how you could use it for your project. So for some background, Facemap basically follows up on previous work by Stringer et al. Study 2019, where they recorded from thousands of neurons in mouse visual cortex and also other brain regions using neuropixels, as shown here, and then using raster map to sort neural activity over time. And what they were looking at is this activity in visual cortex was recorded in complete darkness. And simultaneously, they were also recording mouse's behavior, including body movements and facial movements. And they used this SVD or PCA analysis to compute motion PCs of the mouse face to, I guess, reduce the dimensionality of this behavior into few dimensions of the behavior PCs and use that to predict the neural activity recorded in complete darkness using a linear model. And so here's what you can see, the prediction using the orofacial movements on test data. And this was about across different mice, about 20-25% of variance explained. So the scientific result was a lot of the activity that was considered noise could be predicted by the mouse's own movements. So the model that I just mentioned that they used was linear model or reduced rank regression to transform like those behavior PCs to neural PCs. However, there were some challenges with like when you're using motion PCs to understand what behaviors are being represented. So as you can see from the first few PCs of the mouse face, it's hard to tell which movements are driving this neural activity or what part of the face. And the other point was the linear model used here does not capture temporal information that might be useful for integrating behavioral information across different timescales. So that's how we started working on my project Facemap. And as Carson described earlier, we used a similar experimental setup where we had these large-scale mesoscopic 2-photon calcium imaging recordings in the mouse visual cortex, and this was dual plane, so this allowed us to capture about 50,000 neurons simultaneously. And similarly, we also recorded mouse's behavior using these face camera, and the mouse was head fixed and running on the spherical ball. Now the question was, how can we capture those behavioral movements without using those motion PCs? So at the time in the field, there were these popular tools called Deep Lab Cut and Sleep that you can use for this key point tracking or pose estimation. So we wanted to develop something that's specialized for the mouse face because a lot of labs are already collecting this data set. However, when you start with any of these projects, you have to define the number of key points in a separate project for every session. So we wanted something that we could easily use across a bunch of recordings that we have in the lab. So that's how Facemap came about, and I'll tell you a bit about the tracker part, and then later on how we used it. So to develop the tracker, we wanted it to be something small that ideally could also be used for online tracking, which we haven't tried yet. So we modified this unit style architecture and used similar objectives as Deep Lab Cut's model, where you're predicting the locations of the key point in a downsampled map and also the probability that the key point is in that region in that downsampled map, and then use that information to project the position of key points in your original image space. And this came after a bunch of trial and error and also benchmarking against these tools. And here's what the tracking looks like on the mouse face. And you can see this is on a test mouse, which I would say is very similar to the other mouse data sets in our lab and the data set we trained on. So yeah, the model was trained on about 2,400 frames, and these are about 15 key points. And so here you can see some of the key point tracking over time for the eye key points, the whiskers, and nose key points. And as I mentioned, with any such machine learning topic, you also have to benchmark against some tools. So we trained deep lab cuts models and sleep models on the same data set and finding the best model and to compare in terms of two metrics. One is how accurate they're in predicting or computing the error, and also how fast the network is, which is the inference speed shown at the bottom here. And so Facemap, with the help of much smaller unit architecture, we were able to increase that inference speed by a lot and also get similar level of accuracy as the state-of-the-art models. So then comes back to the point where we wanted something that we can use across different experimental setups. We asked other labs at Janelia and elsewhere, just reaching out to people at conferences if they can share their data so we can try out on our model on how well this works. And so this is, I guess, the base model performance, our model trained on the 2400 frames on the lab data set. And as you can see, that is still quite off, and it can vary depending on the lighting used in those setups, or sometimes if it's a very different camera angle than the ones that we've trained on. So then we tried to integrate this loop of where you can maybe refine a few key points, some of the predictions from the model, and then retrain or fine-tune the network. And on the right there, I'm showing after fine-tuning in about 10 frames, you can get an average level of error about 2 to 3 pixels on this 256 by 256 frame that seems useful for analysis. So that is the current model setup, where you can use our pre-trained model and then train it on your data set or fine-tune if it doesn't work as well to track these 15 key points. So that was on the Facemap tracker, and now we want to go back to the previous question, as well as in, are these key points also just as useful as the motion PCs in predicting neural activity? So we worked on developing this deep key point model that can take the key points as input to the network and predict the neural activity PCs. So to describe the model briefly, the input layer takes the different key points, X, Y positions. And as I mentioned earlier, with the linear model, we didn't have any temporal information. So the 1D convolution filters integrate some temporal information over time. And after a few latent or hidden layers, and also adding nonlinearity to this model, which helps in the prediction, we get our output neural PCs. And here's an example prediction, here's a test data recorded in an example mouse, and the prediction from our deep key points model versus the previous linear model, as you can see. And to quantify across different mice in sensory motor area and visual area, you can see that the deep key point model had quite a big improvement in the amount of variance explained across the different brain regions and across mice compared to the linear model. So we also briefly looked at, as I mentioned, we were interested in looking at what are those behavioral states that might be represented or maybe related to driving this neural activity. So to look at that, we modeled the deep key point features, or basically the activations of this layer, and using a hidden Markov model and looking at some of the different states, which we were able to visually classify as maybe when the mouse is grooming, it's defined as one state, or when the mouse is blinking. Similarly, there's this running time period, and the duration of those states also vary, or when the mouse was resting. So yeah, that was a summary of the Facemap paper, and I can tell you briefly about how we are using it in the lab to ask some research questions in the field. So there is this question, or in the field for primate literature, is it similarly, like they were asking the question, is it orofacial movements, body movements represented in primate visual cortex? And they find little to no modulation, unlike mice, and they claim that it could be related to eye movements. So in our further analysis, we wanted to specifically focus on eye movements in mice and compare that to the rest of the orofacial behaviors, whether which one is more represented in visual cortex. So to do that, we modified our deep keypoint model to take different inputs. So you can train a model by just taking your pupil position, the XY traces. So this is a separate, it's not from the key points, but the original Facemap where you can track the ROI of your pupil and take the center of mass, which is the XY position of that center of mass here. Or you can take the whole face, crop out region of the eye to isolate any of the orofacial movements, excluding the eye movement, and finally also compare with all the information combined. So we wanted to ask this question of how much do these eye movements explain the variance, explain the neural activity compared to these orofacial movements, and also how does that vary in different experimental conditions. So I mentioned we were doing all the previous recordings in complete darkness, so the mouse's pupil is fully enlarged, so you can't really track its position. So what we did is we used this carbachol agent, which is just applying some eye drops to this mouse's eye, which allows the pupil to constrict for a certain duration of time, about 30 minutes, and allow us to track its position. So in darkness, then we can compare how much does the pupil position explain versus the facial movements. And you can see the pupil position is much less, around 10% compared to the 20% of variance explained here. And the static and random condition here, to explain briefly, but if you have more questions, we can get into more details. Static is just looking at when you're presenting, so those two conditions are in when there is visual input, so there is a visual stimulus on the screen, and the mouse is looking at just one image. So basically, its eye position is coupled with the retinal input coming in. In the random case, it's similar to presenting a movie or random images over time. So you are decoupling your eye position from the retinal input or the visual stimulus. So in both those conditions, you can see that the pupil position variance explained is much lower, which is over here, compared to the orofacial movements when there is visual input coming in. So yeah, just some more example of how we've used the deep keypoint model and looking at some of other research questions in the field to isolate more of like which the orofacial movements are represented. So I mentioned that we developed it as a tool so other labs can use, and it's packaged in this software that's available on GitHub, and the GUI looks somewhat like this, where you can drop your video file and either just process the motion PCs as I had shown earlier, or if you want to track the key points, then you can run the predictions using the base model or go through some of the steps that is outlined in the GUI. And we also have a video tutorial if you'd like to follow to refine the key points. And the traces for the key points are shown at the bottom here, and also example traces for the motion PCs. So yeah, if you guys haven't installed it yet or would like to use it, here's a QR code for the GitHub. And if you run into any bugs, let me know. This was pre-LLM, so it'd be good to debug it more. And yeah, thanks to all the co-authors on the paper. And if you have any questions, let me know.
Thank you, great talk. I have a kind of a stupid question. I know humans and macaques can move their eyes without making any orofacial movements at all. In the mouse, are eye movements tied to also making orofacial movements together at all times, or can they just move their eyes independent of their orofacial movements?
That's a great question. In our paper, we have like this figure 3 that's trying to look at the coupling between the direction of the eye movements and whisker movements. So often when they do make saccades, there is some relationship between the direction, but it's not tight coupling because maybe it's different states where they're moving their eyes and whiskers together and some other times where they're just moving their eyes without much facial movements. So it varies, but with this you can quantify as in what are time periods when they are moving them together.
Well, does it depend on the intensity of the eye movement? Like very subtle ones, maybe they don't need to move their faces, but like, I don't know what the max limit of the eye movements are, but within that given range, maybe stronger eye movements, big saccades, or whatever you want to call it, causes more orofacial movements, and there's a threshold cutoff, and then beyond which.
That's a good question. I don't think I've looked at in the figure. It's more in pixel space how much of the whisker movement for nasal saccade versus temporal saccade, and their range of movement is about 10 degrees, but that would be something we can quantify.
Just kind of a question I've been interested in terms of the benefits of using pupil area versus pupil diameter versus pupil velocity for relating to neuronal activity, because obviously for area and diameter, those are kind of different based on kind of like the starting size of the pupil when like a dilation event or something happens. What have you guys found works or seems to best predict neural activity or relate to behavioral states?
Great point. So I think, in this figure, we did try to add pupil area as well, in addition to the center of mass of the pupil. So pupil area, people are typically considering arousal state, or like when you're mentioning the diameter. Whereas in this case, we were interested in time points when there is a saccade or no saccade time period, and I guess isolate those two different behaviors. But we see, at least when you're able to control for visual input, that they explain similar amount of variance. And in the dark condition, we also have another plot that's looking at which of the neural PCs are best predicted. So orofacial movements, you'll see that even the higher dimensional neural PCs are explained by those behaviors, whereas for pupil area and pupil position, you'll see it's more linear, well, it's mostly the first few PCs of neural activity.
Yeah, not so much on that side. So I'm not trying to like argue that orofacial behaviors aren't important, but just in terms of like using pupil area versus having used pupil diameter or pupil velocity.
Oh, okay. I haven't done that comparison myself with velocity versus the other derivatives.
You can get velocity, the model can learn velocity if it wants to, going through the neural network with the temporal process. Yeah. But we don't explicitly put it in, so you can learn velocity of all of these variables if it's helpful to predict neural.
I have two questions. First is maybe related to Catherine's previous results that she found that the movement rather than the visual stimuli can help predict the neural activity. But here, have you compared, for example, the pupil area and the position versus visual stimuli? How much can they predict the neural activity? Which one is better?
Oh, you mean also add stimulus information on top of the behavior variables? Not in this work, but I would think you would need a different model where you're also fitting stimulus at the same time. So it's not something we've looked at.
Okay.
It's about 2 times more signal variance related to visual signaling than the amount of signal variance related to.
A second question is, here most of the variables are more motor variable, and comparing to this visual stimuli variable, do you find any temporal delays for the neural activity? For example, is the visual cortex first respond to some visual stimuli, and then those motor output is more like a inference copy, so maybe later? Have you tried to compare?
So we have not looked at the stimulus part, but for the behavioral information, the idea is that these convolution filters are capturing that temporal delay, and it's both before and after. So, but specifically, like how much is the delay between the movement, or it's usually over a time scale of 1 second that we found if you were to bin that information, that you get the best prediction in decades over time.
We analyzed it in the previous paper too with EFITs from mixed timelines. It's after, we would predict best prediction is after the behavior, more like after.
Sure.
So I don't know, like when the mouse is looking at the visual scene, how much there is overlap between the whiskers and the screen? Like my question, is it possible that the whisker motion with a kind of bright screen on the background creates this kind of sensory motor feedback loop? I mean, that the facial movement encoded in visual cortex because the whiskers swipe through the visual field, and that's how it kind of creates.
I think to account for that in the best way is why we've kept the dark condition, where you don't have that visual feedback of even the whisker motion. I guess to do it in light setup, you would have to trim their whiskers off completely. Mm-hmm.
Yeah, so more like kind of like a simple question, but some of the I think all of the examples that you showed were like the animal like just passively viewing the stimuli. So have you like tested on some data sets that, for example, have like licking behavior, or like where the animal is, I know, using his paw to like move something, right? So in order, for example, like I would like to use it to like replace deep lab cut on my lab.
So you mean including licking behavior and...
Or hand behavior.
I'm sorry?
Yeah, so licking behavior or like paw behavior. Right?
Oh, we initially had a key point for paw, but because this is just like spontaneous behavioral movements, which is the paw only comes into frame when they are grooming. But if you have a task where like the mouse is licking more, motion PCs would be able to like capture that movement as well. Oh. But yeah, we haven't separated just paw or the tongue movements because it's not in our data set.
Thanks.
Mm-hmm. Okay, thank you.